Okay, welcome to the new section, section 5 of ZOO3649, Evolutionary Genetics. And this section is about speciation. So it's about how new species are formed. And in this section, section 5, and the next section, molecular ecology, section 6, we are going to actually take all the stuff we've learned in the first 4 sections and apply our minds to the real world. Okay? And the 2 parts of the real world we want to apply our minds is firstly this part, how do new species evolve? Okay? This is one of the great goals of evolutionary biology. So we're going to apply our minds to this to this task to find out how new species evolve, speciation. And in the last section, molecular ecology, we're going to apply our minds and all the theories and all of the models that we learned in the first part of the module, and we're going to apply that to conservation in the real world. So looking at conservation issues and how genetics can help there. So, but that is in the last section, molecular ecology. So both these sections are designed to make you use all the information that you've learned until this point, or the theoretical information about populations, trees, and coalescence, to learn and variation, to apply them now to the real world, to see with your own eyes how these things actually happen. Okay? And so the process of speciation is quite a complex process. It's a process that has enthralled scientists and non-scientists since time immemorial. And the first lecture today is going to be from genes to species. So the first ideas, okay, by the founding fathers of evolutionary biology, the first ideas of how the process of a new species is actually formed. Okay, so the changes. So you'll notice that this picture here, and I'm actually very privileged to have my picture so closely in such close juxtaposition to this man's picture, because this man is Theodosius Dobzhansky. And Theodosius Dobzhansky was one of the major contributors to the modern synthesis, along with Wright, Fisher, Haldane, and several others. Dobzhansky was also a major founding father of our current evolutionary thinking, and the thinking in terms of populations, okay, which was the modern synthesis. So using this idea of changes in allele frequencies, okay, basically that's what populations are about, right? How do you know that a population has evolved? Because its allele frequency changes from one generation to the next, okay? So the idea was, how do we go from small changes in allele frequency, tiny, what some people call microevolution, small changes in allele frequency. In second year, you would have learned about populations and population genetics. They would have called it microevolution, okay? But how do those micro changes in allele frequencies from one generation to the next, how can that eventually lead to what some people call macroevolution? So lead to the evolution of different species. So basically, this idea of Dobzhansky's was to link microevolution with macroevolution and show that there's no such thing as microevolution and there's no such thing as macroevolution. It's just evolution, and it all happens at the level of the population. Okay? So tiny bits of changes in allele frequencies over time can lead to the evolution of different species. In other words, there's no real distinction between microevolution and macroevolution. Okay? That is a concept in second year and in high school to allow you to train your minds into populations and trees. Okay? Now that I've already taught you about populations and trees, we can leave those terms, microevolution and macroevolution, we can leave it behind in our cradle. Okay? We don't need it anymore because there's only one evolution. And this is how it happens. All right? So Dobzhansky, and you'll also remember Dobzhansky is the guy who came up with the saying for this module, right? At the start of your course guide, Theodosius Dobzhansky's quote of quotes, and that is, nothing in biology makes sense except in the light of evolution. Okay, so you can't be a biologist if you don't understand evolution. That is what Dobzhansky is really saying. So that is why this 3649 is a core module for BotZoo, because you can't become a biologist if you don't understand this. Everybody must understand this in order to qualify as a biologist. Okay, and that's why 3649 is a core module, and it will always be a core module. Everybody has to take 3649 for that very reason, because nothing in biology makes sense except in the light of evolution. So how did this clever guy, Theodosius Dobzhansky, how did he explain how microevolution leads to macroevolution? Well, this is how he did it, okay? And these are the steps in Dobzhansky's ideas. So the first step is that a mutation will occur randomly, and that is how genetic variation is built up in a population, through random mutations. And remember, we first need variation, okay? Otherwise, evolution can't happen, right? So we need variation. So, but if members of the population, if they only breed with each other, and they don't breed with members of the next population, okay, then the new mutations in my population here cannot be shared with the mutation with the other population. It's only then happening in my population. And because there's no gene flow between populations, okay, if you're only breeding with yourselves in one population, that new mutation that happened in your population cannot be shared. It cannot be given to another population, okay? So that's the idea, that the populations are not sharing genes. They don't have gene flow between them. That is, that point, the second one, is the most important point, okay? Their populations must be isolated in order for Dobzhansky's view to work, okay? In other words, there must be no gene flow. So when the populations are isolated, each population will gradually become more and more distinct from the other populations, right? So imagine this is your population here. You can see where my mouse is there. Here's your population here. At some stage there was a barrier. Let's just say a river started flowing between these, in the middle of this population. And so this side was separated from this side. No gene flow. They cannot have gene flow. Okay? So what happens? Eventually, the populations will become different. Why? Because they cannot exchange genes. So you cannot take this light blue beetle and put it here and bring light beetles to this population. And you cannot bring dark beetles to this population. Why? Because there's no gene flow. Okay? So as that happens for over time, there's no gene flow, the populations start becoming different, right? Some of the new mutations turn out to be, after enough time has gone past, some of these new mutations, because they've never been exchanged between the populations, now we get rid of that river, and suddenly the populations are able to mix again. Some of those new mutations will be incompatible with the other population. Because why? Because they've been isolated for such a long time. The new mutations here and the new mutations here never got a chance to be shared. Okay? And so that's why, after these mutations remain in the population for a long period of isolation, and now suddenly they can come back and there's no isolation anymore, suddenly you find that the mutations are incompatible with each other. Okay? And that is what we mean by the establishment of reproductive isolation. When the populations in this last stage here, you see where my mouse is, they come back together, but what do you see here? they cannot exchange genes. Because why? They have already become too different to each other. They've built up so many mutations of their own which were not exchanged through gene flow. And therefore, they've become different. Now when you put them together, they don't breed with each other. They can't mate with each other anymore because of these incompatible mutations building up in isolation over many, 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 many generations. Okay? And this is the idea of reproductive isolation, where you've been isolated for so long that now you can come together, but you can't breed anymore. Because why? You've already become different species. Okay? And that is how small changes over many, 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 many generations, right, can lead to the process of speciation. A new species can evolve just from gradual tiny changes in allele frequencies, but over a long period of time. And most importantly, isolation. Okay? So this idea of how do you go from small gene frequency changes, allele frequency changes, to species, the evolution of different species, the linking micro and macro evolution. This process that Dobzhansky has outlined here depends on isolation. In other words, here, something has got to isolate the two populations and prevent them from exchanging genes. Because if they're able to exchange genes, what happens? They don't become different, right? They remain the same, as you know from Wright's Island model, okay? If you exchange genes, okay, and you exchange genes over generations, you will not become differentiated. The populations will become homogenized. Okay, so it's all depending on gene flow stopping. All right, so that brings us to the question of how do we recognize a species? Okay, inside one species or groups of species, you can actually see quite a lot of variation. I mean, just look at, okay, there are this Ernst Mayer's classical one species of bird, but on the different islands and different parts of the world, they have a different phenotype. Okay, they look different, and Ernst Mayr saw this even within one species. There's a lot of variation. Just take, for example, our species, humans. We have a lot of different kinds of humans, don't we? We have white-skinned humans, we have black-skinned humans, we have brown-skinned humans, we have even a yellowish-skinned humans, we have tall humans, short humans, you name it. We've got many different variants of humans, right? So even within our own species, we have a lot of variation, okay? So Ernst Mayr thought that this variation could be not discretely distributed, but clinally distributed. In other words, distributed gradually so there's no place where you can say, oh, here's one kind of bird starts and the other ends, and they don't cross this line. It's usually not like that. Okay, it's usually that one blends into another, all right, without a hard break in between. Okay, so basically, it's much more difficult to assess variation within species or in closely related species, where the phenotypic variation is not reflected in the genetic variation, or when you have gene flow. So when you have secondary contact, where two populations which are isolated have come into contact with each other, and now they're exchanging genes. Okay, they didn't obtain reproductive isolation, which means when they come together, they can exchange genes. Okay, and what happens in that situation? So the ones that are born when these two populations meet, they look sort of halfway in between one and the other, right? So if you look at from one part of the geographic distribution to the other, it looks like there's a climb, a gradual change in phenotype. Meanwhile, it's not a gradual change. It's just simply a place of secondary contact. So there are lots and lots of things going on within a species or with closely evolving species, which makes it difficult to define what exactly is a species. Where does a species start and where does it end? Okay? So, how much variation is enough to say, oh, two things are different species? Okay? So, that's why Ernst Mayr came up with the idea of a subspecies. Okay? That's a local population within a species that was distinct enough to have a special label for its own phenotype. Okay, but subspecies, and Ernst Mayr recognized this. Ernst Mayr recognized that subspecies have the same problem as species because uh, species are difficult to um, to define for the very same reasons that subspecies were difficult to define. So even though Ernst Mayr came up with the idea of subspecies to try and explain variation within a species, he found that it was difficult to define what a subspecies was because the idea of a species itself is difficult to define. Okay? Unfortunately, this idea of subspecies has become entrenched into biological thinking, and you see people talking about subspecies all the time. They're given a taxonomic value, even when they're described from a very few specimens, so they're not represented. They can still be attributed to a subspecies, or even if there's no boundary between one subspecies and the next, there's still subspecies that exist. So the subspecies idea is very static. It does not account for the evolutionary forces that are changing allele frequencies. Okay, so the however we try to study biology and species or subspecies or whatever, we always need to make sure that we account for the fact that evolutionary forces are changing allele frequencies. We always have to make sure that we account for the four forces of evolution. That is mutation, drift, gene flow, and selection. Because all three of them are working together to create species. Okay, so that leads us to the question, what is a species? Well, speciation is the evolutionary process by which biological species arise. Okay, so but before we can examine speciation, we have to ask ourselves, what is a species? And there are over 26 or more different answers to this question. Okay, and why are there so many answers to this question? It's because it's a difficult question to answer. Okay, that among the main concepts are the morphological species concept. That is, if you look like a different species. That was you remember this name, Linnaeus. Okay, this is the father of classification in the 1700s. He came up with the morphological species concept, which is if you look like a different species, you are a different species. Dobzhansky came up with the biological species concept. And what does that mean? It means that if you can mate with another organism, you belong to the same species as that organism. Okay? And the biological species concept, it makes a lot of sense, but it doesn't cover all the possibilities. There's the evolutionary species concept, which of Simpson, which says that if you have a different evolutionary trajectory, in other words, if you have a different line of descent, for example, if you reconstruct a phylogeny or a tree, and species have different lines of descent, then they are different evolutionary species. So a different way to define a species. But again, not all of these, not a single one of these species is definitive. In other words, there is always a case where the species concept will fail, including Dobzhansky's biological species concept. Okay? Not one of these concepts is actually definitive. So, in other words, the answer to the question, what is a species? There are many answers, but not one answer is actually the correct answer. Okay? And why is that? Why do all these species concepts fail? There's always an exception. Why? Because evolution is not a static process. Evolution is a dynamic process. Evolution is continuous and it is happening all the time. Okay? So at any given time, every population has either become a species or is on its way to becoming a species. Okay? So this continuity then makes it kind of impossible to say, I'm drawing a line in the sand, and on this side we have species, on this side we don't have species. Because every population is in its own place along that line. Okay? And there's no way to just draw a line and say, above that it's a species, or below that it's not a species. It's impossible, because every population is evolving in its own continuous time. And so it's impossible to draw lines in the sand. Okay? So for every attempt, there's always an exception to the rule. But be that as it may, there may be many species concepts. They might all have exceptions, so that not one is definitive. So we can't actually answer the question, what is a species, perfectly. We can't. That's one of the problems in biology. We can't actually answer that question because of all the reasons I've given you above here. But there's one undeniable fact. Species do exist, right? We can't define what a species is perfectly, but we know that there are species. We humans, we're a species. Dogs, they're a species. Cats, they're a species. Trees, how many different species? Thousands of different species of trees. Thousands of different species of beetle, as you know, because you've been working in animal phylogeny with Dr. Skuman, right? So there are thousands of species. We know that there are species. We just don't know how to define them perfectly, all right? We see species everywhere we look. We ourselves are a different species to chimps and gorillas, okay? We're not the same as them. We're closely related to them, but we're not the same as them. So we know that there are species. Any fool will be able to look around and say, yes, there are species. But when we try to define what a species is, that is where we run into problems, because evolution is a continuous process that is happening all the time. Okay? And that leads me to the next lecture, which is outlining the classical view, the Dobzhanskian and Mayerian view of the speciation process. And that will be in the next lecture.